Hi, in this video we're going to talk about the antigen and antibody interaction. Let's get started. Let's start off by discussing what an antigen is and what an antibody is. An antigen is any substance that elicits an immune response. Specifically, this is going to be the production of antibodies. Antigens can be parts of microbes, for example, the flagella of a bacteria or the capsule of a bacteria, or they can be products of microbes, for example, toxins created by a virus. They can also be non-microbial, meaning pollen or egg proteins or donated blood cells can trigger your immune system to produce antibodies. In this diagram that we have here, we have a bacterial cell and its flagella. So in this image and throughout this video, this is the example that we're going to be using. And in this case, the antigen here is the flagella. An antibody is a Y-shaped globular protein that binds to the antigen that it is specific for. In other words, it is a Y-shaped globular protein that binds to the antigen that it was made in response to. Because an antibody is a globular protein, it's also referred to as an immunoglobulin. Now that we've discussed what an antibody and an antigen is, let's start to talk about their interaction. Here we have our bacterial cell with its flagella or tail-like appendage, which is going to serve as our antigen in this case. And as you can see on the flagella, there are three different epitopes or specific regions that antibodies can bind to. So in this image right here, we have the white antibody being able to bind to the triangular shaped epitope. However, this white antibody would not be able to bind to the blue and yellow epitope because it is not specific for it. However, these antibodies, the blue antibody can bind to the blue epitope, and the yellow antibody can bind to the yellow epitope. In real life, an antigen is likely to contain many epitopes, and so there's going to be a different antibody that can bind to each different epitope. Once an antibody binds to an antigen, there are several different things that can happen. And these things include agglutination, the neutralization of the pathogen, the opsonization of the pathogen, the activation of the complement system, and antibody-dependent cell-mediated cytotoxicity. In my next video, I'll be talking more about what happens in each of these events. But for now, that's it for this video. I hope it helps. Please like, subscribe, comment, and share.